So, like Elena said, a lot of my research involves evolution of features in mammals. I'm interested in the headgear of things like deer and antelope, but I'm also interested in the teeth of all different kinds of mammals, including horses. And early on in my career, it became clear that there were a lot of other people who collected data that I could use to advance my research. And I had to go through a lot of old papers, copying data tables out. I had to go through old papers, copying photographs and then reusing the photographs to measure shapes and angles on specimens. And it just felt like there ought to be a better way. And it turns out I wasn't the only one thinking that. So that's where the whole field of bioinformatics has come from people wanting to be able to share and reuse data in a better way. And that's what I'm going to talk about today, is some of the efforts that we have been making as a global research community to make it possible to find other people's data and use them to answer your own research questions without having to do a lot of extra work. And then also, when you collect data, make them available so other people can discover things you didn't even think about when you were collecting this data. And I think that that's going to facilitate a lot of work moving forward. And in particular, it's important. Let's go back one. It's important to me in terms of understanding how animals and plants and all kinds of organisms are responding to climate change. So in the end, a lot of the research that I've been doing with other people's data is about the shapes of animals, but in particular, the shapes, how those shapes evolve in the context of changing climates, either today or in the deeper time past, okay? And so I was going to start out with a couple of the projects I've done looking at animals moving around, having their geographic ranges change in the context of climate change, but I did it using data that other people had collected and I didn't collect directly. So this map is from my postdoc and the paper that I published from my postdoc back in 2008. And what it shows is actually a map of species endemism in California. So it's not just species richness. The areas that are brighter colors, brighter red on this map, aren't just spaces that have more species, they actually have more younger species. And so what we did was we went through all of the different species of mammals that are found in California, and they all had genetic data already available on GenBank. So I was able to go through, as a paleontologist, I never touched any living animals, and I never extracted any blood or other samples. I never did any gels to get DNA data myself. What I did was I took everyone else's work that they had done for their own research questions and put on GenBank, and I extracted those data and reorganized them to answer this question. And I was able to make a map so that we could see the areas in California that had a lot more younger species in them. And the idea was that under models of climate change, as species are going to rearrange themselves, we wanted to look for areas that were going to have a lot of relatively young species because they represented places where speciation had already been happening over the last 2 million years or less, okay? And in particular, we were trying to understand this area. It's the confluence of 5 different ecozones. You've got desert mountains, you've got the high mountains here, you've got the coastal mountains and the coastal valley here, and even these different coastal mountains coming together. And so we were able to see that there were many more young species there than you would expect by random chance, and that made it into an area with a higher priority for conservation for the state and for conservation groups like Nature Conservancy. Okay, and then this study is one I did with my colleague Jenny McGuire, and it's actually mostly her work. So when I talk about this one, this is mostly what she did. But for this project, we were trying to understand microtus, so voles, kind of rodent. We're trying to understand how their species distributions have changed with changing climate from the last glacial maximum to today in an effort to understand whether we could project from today into future climates and be confident that they would continue following our predictions of species range changes. And so Jenny actually went through the collections at the museum in Berkeley and also I think several other museums and very carefully identified all of the fossil specimens from individual teeth. These are, mouse teeth, so they're microscopic. And so all that work was individual work that she did, but then eventually becomes the sort of data other people can use in the future. But for the modern distributions, we pulled data from the Arctos database, which is I'll talk more about this. It's a shared database of museum collections from across North America that lets you go in and pull out specimens of modern animals that have been collected. And so those modern animals, we were able to use their data to create ecological niche models and then project where they would like to live in the modern climate based on the temperature and precipitation and other environmental variables, and then take that information and project it back onto a model of the last glacial maximum just to see whether or not those same species are found in the fossil record in the places that we would expect them to if they were living in the same kinds of habitats that they like today. And some of them did a better match than others. So there's some of them that we felt like you could be more confident, like Microtus californicus, projecting forward into future climates, when some of them did a much poorer job, like poor Microtus townsendii, which seems to have had some kind of a shift in its ecological niche between the last glacial maximum and today. So if you're going to try to project where this one's going to live under climate change, we want to think about other variables to include in that analysis. Okay, but in the end, right, these data were data that Jenny actually went and spent time on and touched the specimens, but these were all data that we pulled from the database. And we did do data cleaning. That's an important step I'll talk a little bit about too. But those are just two examples from my research career of places where I've been able to build big projects without actually touching the data myself. And what we want to do is make it possible for more and more people to be able to do that kind of work. And we still want people to go out and collect new data. That's important too. But one of the problems that we see, at least in the United States, and this may be a problem in Europe too, is that people who go collect new biological data have a hard time getting funding to do that work. The funding is mostly available for big projects that look across a lot of data. And so one of the things we need to do is a better job of tracking the use of individual data sets so that we can go to the funding agencies and say, look, this one expedition that costs not very much money actually has contributed to these hundreds of different analyses and is critical to do all of these different kinds of expeditions to be able to build the edifice that we need for the larger scale studies. So tracking data use and being able to document the importance of individual field expeditions is another benefit of having this kind of integrated biodata. Okay. Oh, actually, let me put the chat. I don't want the chat. Go away. There we go. One more. Okay. So in the end, the big question that Elena asked me to answer with the talk today is how do we preserve and share the huge amount of data that are preserved in all these collections? And there's a conservative estimate that there's over 1 billion specimens of modern animals and plants collected in USA natural history museums, okay? So just in the United States of America, the museums have over 1 billion of these objects that need to be cataloged and shared. How do we do that? And there's two key concepts that I want folks to take away from today that I think are critical for the future of this kind of bioinformatics work. Now, one is the concept of the extended specimen, and the other one are FAIR principles: findability, accessibility, interoperability, and reuse of digital assets, okay? So what's an extended specimen? So the extended specimen concept is one that starts with the actual physical specimen. You go out in the field with a trap, you trap a mouse, you humanely kill the mouse, and then it turns into a skeleton and a skin that you have in the museum collection. Maybe you also preserve the gut contents, okay? Or you go to the field, you collect a plant, you press the plant, and it becomes an herbarium sheet. That's the core of the physical specimen, but there's all kinds of other things that radiate out from the individual physical specimen that we have to account for in the structure of what we're now calling the bioinformatics ecosystem. Okay, so there's all these different kinds of data that are generated from individual specimens that have to be accounted for and organized so they can be discovered. And this is a principle that I think I need to reiterate multiple times, is that whenever you have data, you have metadata about that data. So whenever you have an observation, then there's other information that describes the context of that observation. And metadata at one level is always data at the next level up. So what you consider metadata about your specimens may be actually the fundamental building blocks of someone else's analysis. And so they need the metadata about your metadata. And so all of that has to be organized and controlled in a way that we can keep track of, and preferably use computers to do most of the tracking for us, because there's so many specimens, there's so much metadata that we can't possibly as human beings track it all ourselves. Okay, so just to kind of really illustrate this. So you start with a physical specimen like this mouse skeleton that was collected in the field. And I'm actually kind of cheating, because one of the things that's created from this mouse skeleton is a primary extension of specimen media, photographs of the specimen, right? And so that's what this actually is that I'm showing you here, is a photograph of the specimen, not the specimen itself. So that's actually part of that primary extension. And then you also get the digital specimen record. So this is a Microtus californicus specimen from the Denver Museum of Nature and Science that was collected in Oregon. Now this specimen lives in Denver, Colorado, okay? But if you wanted to actually go visit, you could, because you have the metadata to tell you which exact specimen to go back and look at. And if I go to the museum and I start measuring things from it with my calipers, or if I'm doing geometric morphometrics and I'm using the images to digitize landmarks, then that actually becomes part of this secondary extension, where I'm building secondarily on top of even the images and the location information. And then when I go to the step of, like I did in that paper with Jenny McGuire, building geographic ranges, and we're actually up here in this tertiary extension where you've got geographic ranges, you've got summary trait data, what's the average mass of a Microtus californicus, that's out here in this area. You're building a phylogeny, that's also out here in this area. Okay, so these are all nested parts of the extended specimen. And so the only thing that is just data and not metadata is the specimen itself, but everything else is metadata at one level and then data at the next level. Does that make sense? Okay. Let's make it safer to use the keyboard. Okay, so the next thing I want to talk about are FAIR principles: findability, accessibility, interoperability, and reuse, which are critical kind of parameters for how we want these data resources to work into the future. And none of the ones I'm going to talk about today actually hit all of the FAIR principles yet, but we're all trying in our different corners of the ecosystem of bioinformatics to reach this spot. Okay, so this is sort of the castle at the end of the road for us. So for findability, we want every object and every piece of metadata to have what's called a universally unique identifier, or a UUID, okay? So universally unique identifiers are like serial numbers that are given to data, observations, or metadata that are also resolvable on the web. So if you type it in, it'll actually take you to a web page that directs you to the electronic record for that thing in whatever database it lives in, okay? And so one example of these UUIDs are the DOIs that you get for scientific papers. So if you have a scientific paper and you have the DOI, you don't have to type in the whole reference. You can just type the DOI into your URL bar, and it will resolve to the actual reference itself. So with UUIDs, it's the same kind of a deal. If you have that UUID, you can get back to the original specimen record. We really have to have clear identifiers that link the metadata and data back together. If you have these UUIDs, they can serve that purpose. And then your data have to be indexed in a searchable resource. So this is the difference between taking a data table on your paper and putting it into something like Dryad, where it is freely available and it's archived so that it'll be available indefinitely, and putting it into something that's more like GBIF or the Neotoma paleoecology database or the Futures database that I'll talk about later, is that if you put it in a data table in a repository like Dryad, then it's not really searchable. You can't find the individual records. If you find the paper and you know that that paper has observations that you need, you can go download that data table again, but you're not actually finding the data originally. You're finding the paper and then going back to the data. So that's what we want, is to find the data without ever seeing the paper. The data have to be accessible too. And that means that whatever resource you're putting them into can't have some specialized retrieval protocol. It has to be something that's open and available, like an FTP or an HTTP. So it's not helpful if you put your data up, but it's only accessible through some proprietary protocol. And then the metadata have to persist indefinitely, even if the original data are lost. So you have to have a way that when you're building this resource, the metadata will persist into the future, even if the original specimens are destroyed, so that people can still go back and see that those things did exist at one time and use that information in their research. Okay. They have to be interoperable, okay? And that means that when you're modeling your data, the information you're putting into your spreadsheets, your accessible records, have to be built in a commonly used form and vocabulary. So this is where community standards come in. We have, lots of people working together to brainstorm up the standards that we use for communicating these ideas. This is where you get things like Darwin Core and the ABC that I'll talk about in a little bit. I want to emphasize that when we're talking about this, sometimes we'll say that they have to use a common language, but what we're not saying is that they have to use a common human language. They have to use a common scientific language where we are agreeing upon a standardized terminology, and it might be in Latin for all that matters. Ideally, the standardized form isn't something that humans can read, it's something that computers can read, so that we can automate the process of exploring the data and finding resources that are appropriate. So that's where starting to move into what we call ontology language is important. And I'm going to talk about that more in a little bit too, so that the computer can actually reason over the data set and make connections between different concepts that no human being maybe necessarily has made the connection directly before. And then you have to make sure that if you've got data that connects to other resources, those resources are mirrored in both of those data sets. So if you have a specimen that has a DNA sample that's been sequenced and is in GenBank, then the GenBank record needs to point at the specimen record, and the specimen record needs to point back at the GenBank record, so you can find it from either direction. Okay. And then they have to be able to be reused. This part is basically just about having really rich records, putting in as much information as possible. So instead of just going through and doing the minimum requirement for whatever data resource you're building, actually making sure that you fill all of the fields that are possible to fill for those records. Including detailed provenance. I mean, this is something I think is really basic to all biologists and paleobiologists, is that you have to know exactly where it came from and when it was collected, and who picked it up, so that you can put that story back together again. And then again, you have the community standards showing up. It's really important to have community standards for what are the minimum data that you should include, but also for how to encode the data, right? And so I just put all of them together on a single slide also, so that people who are flipping through can actually freeze frame it there and then hold the slide if they need to have it as a summary. And I'm pulling all this from the Wilkinson et al. 2016 paper. So that's a really good resource if you want to know more about these ideas. Okay. So I mentioned that interoperability requires some kind of standard protocols for communicating your data. And there's two main protocols for biocollections data right now. And they're very similar, and they're actually both standards of the same group, TEDWG, TDWG. TEDWG used to stand for the Taxonomic Diversity Working Group, but they changed what it means to Biodiversity Information Standards. And it's one of those things on the web where they purposefully have an acronym that doesn't match the words that it stands for underneath. But TEDWG is a scientific society that has meetings, I think, at least once a year, where they talk about the standards for how to share bioinformatics data. And whenever there's a change proposed, it has to go through a whole process with committees to make sure that the change isn't going to disrupt existing data and is compatible as we move forward. So the two main schemas that they have are the Darwin Core schema, which is one I'm more familiar with, that uses simple text or XML or the RDF formats to encode the data. So RDF is a way that you encode data for ontologies, which I'll talk about when I get to the FUTRES database. And it's primarily used for standardized extracts of museum collections. So our museum at the University of Oregon has a provider where we take all of our museum data, which are in our own format, and we turn it into a Darwin Core database and push it up to the web so that GBIF and IBIGBio can pull those data out. There's another format, which is the ABCD, Access to Biological Collections Data Standard, which is very similar to Darwin Core, but different because of the way it's being used. And it is, as far as I understand it, the backbone of GBIF, the Global Bioinformatics Facility, and BioCase, which is a consortium of museum databases that's focused in Europe. Okay, so they're using this as the structure underlying those actual databases, as opposed to Darwin Core, which is more of a structure that you use to repackage data so that it's readable. So they could think of this as more like a standard architecture, and this is more like that adapter that takes you from the North American plug to the European plug. Okay, both of these are important for making data interoperable. And then I'm going to talk about some specific bioinformatics repositories, and I'm going to list as many as I was able to think of, but I'm probably going to miss a few. And then I'm going to go through and get some examples from the ones that I'm most familiar with, but they're not supposed to be comprehensive. And I think there's a really important kind of basic dichotomy in these bioinformatics resources between the ones that are really focused on neontology, that's what paleontologists call modern biology, we'll call it neontology, as opposed to paleontology. Okay. And so the modern biology focused databases often do have some paleo records mixed into them, but they're focused on museum collections. So they're focused on the specimen level, the individual objects that were collected by researchers and put into museums. And that's a difference from the paleontology ones, which tend to be focused on published records that have gone through the peer review process. And so they're centered around what we call an occurrence, which is the intersection between taxonomy, space, and time. So you know that this species was present at this place at this time. And you can have multiple specimens in an occurrence. Okay, and so that's a difference that actually is important, not only conceptually, but also structurally, because it affects the way the databases are designed, and that affects the way that you can align the data from the different sources if you're trying to use them for a study. The data from the neontology focused data providers tend to be relatively unclean. They tend to be what the museums have in their collections databases. And so it might be that some first year undergraduate student was paid to go through and identify specimens, and they were at the beginning of their learning experience, and so those identifications might not be as solid as ones that were made by like a third-year PhD student. But you won't know when you're downloading the data who it was that did the identifications that you're dealing with in that case. So it's often useful to do a little bit of detective work to try to clean the data, actually coming from both of these resources, but these often need a lot more care taken to find those kinds of outliers that were introduced by a lack of expertise or miscommunication between the field workers and the collections workers. There's a lot of things that can go wrong that tend to get cleaned up when researchers go through peer review, but that hasn't happened with these. These have been through peer review mostly, which means that you've had people independently looking at the data sets and saying, actually, I think there's a problem that you need to rectify before it gets published. But you still need to check those data. And the need to check the data, that's part of why we need to move towards these FAIR principles, because if we get to the point where all the data really have been checked or problematic data are flagged ahead of time, then that'll simplify the data cleaning and reuse process for researchers. Okay. So, what are the major bioinformatics resources that I think of when I do my research? For the neontology focus, the biggest one is GBIF, the Global Bioinformatics Facility, which is European-based and actually really is the best resource to go to first for finding comprehensive bioinformatics data. BioCase is important, but it's focused on European museums and European collections, so it hasn't been one that's been a major part of my research so far. iDigBio is sort of the North American response to GBIF that was funded by the National Science Foundation and has more of a focus on photos. Canadensis is the Canadian equivalent of iDigBio. A lot of folks don't know about that one, but it can be a really important resource, especially if you're focused, if you have research that takes you into Canada. Arctos is a database system that's actually very similar to BioCase, but for North American museums. Morphosource is a place where people can put 3D scan data. And actually, I know a group of people who are working on a grant to be able to actually encode 3D landmark data on the scans in Morphosource. So that's a resource that can be useful if you're working with imagery, especially 3D imagery of vertebrate organisms. And then Futres is a database that I've been working on that I'll talk more about, that's focused on measurements of organisms and making it possible for people to find and automatically align measurement data between different kinds of vertebrate collections. The paleontology focused, the paleobiology database is kind of the granddaddy of paleontology databases. So that's the place to go first for looking for paleontology data. But then if you're looking for stuff that's younger and you want richer data information in the Atoma database, the NOW database is one that's focused on Europe, but does have North American and African data. And then Open Context is one that is focused on archaeological contextual data, but has a lot of biological data in it too. Okay, so now I'll walk through these different resources. And because I'm here in Venice, I went ahead and explored what kind of records each of these resources has for the area around Venice. And I was kind of surprised, and maybe you will be too. So GBIF is a global initiative. It's located in Europe, but it's accessible to everyone in the world. And like our data in Oregon, it's sent to GBIF and are indexed there and visible through their portal. It has open access to all data. I should have put, it also has an API. So an API is an automated programming interface. And that's a way that you can access data not by a web portal where you as a human being are typing things into boxes and clicking on maps, but instead you can write code in R or another language where you actually tell the computer to go and access the database directly, and it can make much more powerful queries than we can set up for people to use through web interfaces. So web interfaces are a good way to just poke around a little bit and see if there might be records that you want, or they're really good for classes where you're trying to teach people how these resources work. But when you're actually doing the science, you'll want to try to use an API so you can write code to go and dig into the database in a really much more powerful way and pull the data directly into your research script in a format that you can go ahead and analyze right away. There's more than 2.2 billion records in GBIF. It's kind of crazy to think about. It has metadata to talk about the occurrence of the organisms and also the original collecting information. So you can reconstruct a lot about the context of the collection of that organism. Went ahead and looked at the area around Venice, and I found 41,000 observations in GBIF just for the area that I've shown in this little map here. And what's interesting is that most of them, 38,000 of those observations, are iNaturalist observations of uncollected organisms. People seeing birds, seeing mammals and fish, seeing plants, recording them in iNaturalist, and then those get uploaded to GBIF. So those are good because you can use them for research to understand distributions and some things about activity during the day and night, some things about timing of flowering for plants and things like that. But you can't go back and take DNA samples from iNaturalist observations. That's the one disappointment. There's 2,300 preserved specimens from GBIF in this area, and several hundreds of those, I couldn't get a precise number, but several hundred of those specimens are actually at museums in the United States and not in Europe. So if you're doing research on the area around Venice and looking at modern organisms and their distributions over the 20th century, there actually are several hundred specimens that are in North American museums that might be pertinent to your research and would be worth investigating. And then in GBIF, there are 17 fossil specimens from this area. 15 of those 17 are in museums in the United States. And most of those were actually in the museum at Berkeley where I did my PhD work. So at some point in the 20th century, some scientists from UC Berkeley came to Venice and collected specimens, and now they're in the museum in California. Okay, most of these specimens were invertebrate fauna from the Cenozoic, so shells of marine invertebrates. Now, iDigBio, like I said, is sort of a North American response to GBIF. It's funded by the National Science Foundation, and the physical location of the resources in Florida, it's sort of shared in support between the University of Florida and Florida State University. And the University of Florida has one of the most amazing museums in North America, at least from a natural history perspective. If you ever get to go to the US, I actually recommend going to the University of Florida Museum and working with the collections there because they are amazing. There's open access to these data. They do have an API, so you can dig into them directly with your code. And there's a focus on images as well as specimen metadata. So whereas GBIF is all metadata, descriptions of specimens, where they're located in museums and where they were collected, iDigBio also has photographs. And that was a big push of the funding, was to digitize not only the metadata about collections, but also to take photographs of them so people could see what objects were in the museums and get access to them. And there's over 131 million records in iDigBio right now. And when we look at the area around Venice, we see that there's 1,060 specimens in the iDigBio database. These are all North American museums, right? So there's 1,060 specimens from the area around Venice that are physically located in museums in North America. And 160 of those have images, and 14 of them are fossils, and they're not the same fossils as you saw in GBIF. Some of them are the same, but not all of them. And so this is just a snippet of the array of photographs of Venetian specimens that I could pull up on iDigBio. And what you can see is the vast majority of them seem to be herbarium sheets. But there also were a lot of entomological specimens as well, butterflies and many ants. Just when I did my really brief scroll through what was happening. And a lot of these collections, they photographed the specimen cards too, so that you can get a direct look at the collecting data that's on the specimen card, because it's not always transcribed properly into the electronic metadata. So that's a backstop. Okay, so that's another resource for people to look at. FutureS is my pet project. I've been working on this since 2018, I suppose. And so FutureS is a new database that's focused on measurements of specimens. That's why we have this logo that the F is measurements on the skull. It's focused at the specimen level too. So a lot of the places where you can get measurements of organisms on the web are species averages. You get the species average mass or the species average snout to vent length. And we wanted to be able to look at individuals and see the variation within populations and also across the landscape and through time. And so that's the focus of this database. Specimen level, the data are freely available, and we have an API so you can access it. In fact, the API is much better than our web portal, which is still really a prototype. And the thing about FutureS is that we based it on an ontology system instead of on a relational database system. So instead of having the data recorded in individual data tables that are linked together by key fields, the data are actually all modeled in a knowledge base. And that was important because as we add new measurements, we didn't want to have to create new columns in a database and make the thing become unwieldy in its size with all of the empty space it would have. And also, by making it based on ontology, we have been teaching the computer to understand vertebrate skeletal anatomy, so that if you have questions about, I want to find measurements that are from arms, from forelimbs, you can ask it that, and it will bring back all the measurements that are from any elements that were in the forelimb. Okay, so you don't even have to go through and do the detailed work of listing all of the different elements that it might look for, because the computer understands that a forelimb includes all of these different elements. And as we build this ontology, we're actually building it on top of work that's been done for biomedical research. So all of the things that we're doing here are going to be forward compatible with work that people are doing on evolution of development and genetic controls of the skeleton. So I think that's going to be exciting too in the future. We already have 2.4 million records. Most of those come from modern organisms that were in iDigBio, where people had recorded measurements like lengths and masses, but there's not a spot in the iDigBio schema and the Darwin Core schema to put those measurements. So they had put them into the sort of other field. And then one of my colleagues, Rob Brelnick, had students write computer code that could actually extract that from what was a human readable form and turn it into computer readable that we were able to put into the ontology database. Okay, and so what is an ontology? This may be review for some folks, but basically, instead of having the data in a spreadsheet, we're modeling it as a subject verb object connection of ideas. And so we're teaching the computer how all these ideas connect, and that creates a graph network of knowledge that the computer can then use to find connections. So you might think of it like this: you have a specimen as a subject, then it might have the verb of it has a mass of, and then the object would be 10, the number, but then you would need to have that have units of tons. So then you know this specimen weighs 10 tons. Okay, and then you might say the specimen belongs to taxon and have Elephas maximus, right? And then in this case, it actually has a name. This is Tusco the elephant, which is a specimen that's located at our museum in Oregon and died June 10th, 1933. So we have the context of when it died and where it died as well. This is a kind of a silly example, but it illustrates the idea that what we're doing is we're mapping the concepts of the specimen's information instead of just building a spreadsheet where the human being knows what the labels for the columns are. Instead, the computer has some sense of how these things are connected to one another by the graph network that we're building. Okay. But we don't have a fancy map yet for Futros, so I can't show you a fancy map. And we don't have any Italian data, so I can't show you any Venetian data for Futros. So now we'll jump into the paleo data realm. The paleobiology database was one of the first ones to start putting data together this way for paleontology. The initiative was started in the USA, but it's really global in scope. And as you can see from this map, there are specimens, there are occurrences in the paleobiology database from over the entire world. And I'm not going to show you this today, but you can go play with their tools on the website. They actually have all of the ancient continental reconstructions. So when you're looking at Miocene data, you can tell it to move the land masses back to where they were in the Miocene. But if you're looking at Cambrian data, you can have it move the land masses to where they would have been in the Cambrian, which can be really useful. But also, this is cool. These data are also available through an API. It focuses on occurrences. Taxonomic data also has good bibliography built into it. So it's a good way to find references to read and learn about different areas you're trying to work on. And it has a huge stratigraphic data database built in too. The ages in the paleobiology database are recorded in geologic timescale units. So it doesn't have the ability, or at least it didn't the last time I checked, to do exact numerical dates. So it's all based on relative age dating into geologic timescale units. And there's over 1.5 million occurrences in the database. When I looked at Venice, I only found 22 occurrences in the paleobiology database. So these are from publications. One of them is a Miocene fish. That's a kind of pupfish. And then 21 were Holocene fish and reptiles that came from Tenuta Zuccarello, which is a Roman well that Elena actually is familiar with the site, right?
Oh, I'm not familiar. I just check online.
Oh, okay.
It's northern airport.
Right. And so this is, I guess, technically an archaeological record, right? So the well was dug in Roman times, and then these animals fell into it and were preserved. But the records are there for people who want to understand it. Okay, the Neotoma Paleoecology database kind of complements the paleobiology database because it's focused on younger intervals, and it's focused on sites that can be dated more precisely with radiometric dates. So using numerical dates instead of relative dates. It has an API for accessing the data. Again, it was started in the United States, but it's global in scope. And in fact, it includes the European pollen database. So most of those sites in Europe are actually fossil pollen data. And there's over 1 million occurrences, but I need to talk to the Neotoma folks because it's really hard to tell exactly how many they have. And that's something that you should be able to brag about. When I looked in Venice, there are 2 sites in the Neotoma Paleoecology database from Venice. They're both sites from the European pollen database. And so they represent pollen records from cores that I think were taken in the lagoon. And unfortunately, they were put in kind of as stub records, or I guess when they were collected, there was not, absolute dates were not assigned to the layers in the cores. And so the core data can't be plotted with abundance versus time plot, which is one of the cool things that this database will do for you automatically. So that's what you kind of see right now. It's just a list of plant taxa and then pollen counts from different levels in the core. The NOW database is a database that started focused on Europe and then has expanded data to North America and Africa. It's also focused on Cenozoic mammals, okay, which is my specialty. So I'm excited about this database and use it for some of my work. And it is able to do both absolute numerical dates and relative dates. It has over 71,000 occurrences in it. And when we look in Venice, all we see are 2 species of Leptobos from Alante from the Pleistocene. And this is one where you told me that this is wrong.
Yeah.
This pointer should be
Over
over
there.
here.
Yes.
Yeah.
It's very-
So we'll put in a report to them and ask for them to move it to the correct location.
I think you're done.
Mhm. So,
Very good.
and then the last one I was going to talk about is open context. Open context, like I said, is focused on archaeological data. And so it is a place for people to put their archaeological data and make them fair, so accessible and reusable. But and they have an API, so the data can be accessed directly from the computer. And it does include a lot of archaeobotany and zooarchaeology data, but most of it is actually not. Most of it is the human contextual data. And so while there's 2 million records overall, only 500,000 of them have some kind of taxonomic identification. And so our zooarchaeological or archaeobotanical data. When we look in Venice, we see only one site at Cornuda with 4 auroch specimens. Okay, which is interesting to me because this is a different one than we saw from the other one. So far, none of the paleontology databases have really shown the same specimens. So if you were trying to understand this area paleontologically using resources from the web, you really would need to go to all of the different resources because different ones have gotten different records from different publications and they're not all together in one spot, which is in the end part of the point of my talk is that when you're doing this kind of work, you really do have to go across all of the different resources and make sure that you're finding the data that you need and then checking them against one another to look for duplicates because sometimes they do happen, but not in this case. Okay, so in conclusion, as we move forward with bioinformatics, we have to keep in mind the extended specimen and think about how physical objects lead to all kinds of metadata that we need to keep connected so that we can discover the data, but also discover the biological connections that are what we really care about. And we have to keep in mind fair principles so that our data are findable, accessible, interoperable, and reusable. So that as we contribute data, other people will find them and build on them. And also when we have questions we can't answer with just data we've collected, we can find and reuse those data. Eventually, when these are fully implemented, we'll have automated data cleaning. So that'll save us a lot of time and make it so that we can spend more of our time thinking about the actual science part and less of the time sitting in front of the computer, clicking through rows on spreadsheets, making sure the data don't have mistakes. But until then, you have to do that. Many US institutions do hold specimens from European localities. So if you're interested in understanding how things have changed their distributions in Europe over the 20th century, it would be a good idea to check with US collections as well, because they may have some key specimens for helping to understand that context. They may have, if you're doing paleontology research, they may have some key specimens you need for your paleontology research too. If you collect new biological data, make sure that they do end up in one of these open repositories so people can find and use them. And then in the end, why are we doing all this? Because we want to be able to make the world function as the climate changes. At this point, I don't hold on any hope that we're going to stop the climate from changing, but as the climate changes, the organisms change, the organisms move, and they reorganize their ecosystems, and we want to be able to make sure that we are managing the lands in a way that will keep those ecosystems healthy. And to do that, we really do need a fair data. Thanks.